to the Mind and Matter podcast. I'm your host, Nick Jikomes, and today I'm talking to Dr. Carolina Tropini. Dr. Tropini is an assistant professor in the Department of Microbiology and Immunology at the University of British Columbia, where her lab studies the microbiome. And so we talked all about the microbiome. There was some good discussion near the beginning about how the microbiome gets started in our bodies when we are babies, how the microbiome is seeded in large part by the mother's microbiome, the vaginal microbiome that the baby passes through when it's being born. We talked about how the microbiome is important for an infant's ability to digest certain unique compounds, certain nutrients that are present in breast milk. We talked about the differences in the microbiome that are observed when babies are born the traditional way versus through something like a C-section. And then we went through the different parts of the digestive system, from the mouth to the stomach and intestines, and talked about how the microbiome actually differs in these different places. How is the microbiome of the mouth different from the microbiome of the colon? What types of bacteria live there and what are they doing? How are they adapted to those little environments within our bodies? We talked about everything from feeding the microbiome, things like fiber and what that does and what the microbes produce from that our bodies then use. We talked about how our diets today differ in their fiber content and other nutrients from traditional hunter-gatherers, the types of people that we're descended from. We talked about gut issues and gut diseases like inflammatory bowel disease. We talked about things like probiotics and antibiotics, how these things do or do not impact the microbiome. You know, what happens if you have to take antibiotics for an illness? What happens if you take probiotics? Are the probiotics that are marketed to consumers very much these days, are those actually likely to do anything of benefit for you? We also spent some time in the middle or towards the end talking about microbial endocrinology. So that's the field looking at how microbes, such as those found in our gut, actually influence things like sex hormones in our body. So microbes can actually produce or modify things like estrogens and androgens like testosterone in our bodies. And that was just all in the context of the wide-ranging influence that the microbiome can have on our biology. So if you're interested in those things, this will be a really interesting episode. We covered lots of interesting stuff in this one. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it with the first meal of each day, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure. And vitamin D is super important for the proper function of the immune system and for a variety of other things. And there's even evidence indicating that vitamin D deficiency is correlated with more severe cases of COVID-19 in those who get infected. Every time I go into the doctor each year for a checkup, I'm always told that vitamin D deficiency is very common and I should be supplementing on a daily basis. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase. Hey everyone, I want to take a minute to tell you about a really cool health monitoring device I've been using for several weeks. It's called Lumen, and it's a handheld, pocket-sized device with a sleek design. It measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs. It improves your ability to burn fat, which decreases your hunger levels and makes your body less dependent on snacking, and it can increase your energy levels by helping you develop a high-functioning metabolism. I use this device in the morning, before bed, and after meals and workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I need to fast each day to promote fat burning. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. The easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MIND, M-I-N-D, in all capital letters to get $50 off your Lumen device today. And with that, here's my conversation with Dr. Carolina Tropini. What did you eat for breakfast?
I ate cereal.
What kind of cereal?
Well, so it's kind of a loaded question because this is a strong area of negotiation with my family about which cereals are going to be allowed. So we do 1 scoop of cereal that has a little bit of sugar and 1 scoop of cereal that doesn't have sugar. But some kids in my family cheat and get more sugar than they're supposed to.
I see. And how much of that is intentional given what you study?
Well, to have cereals with high fiber is very intentional. So we try to load up our diet with as much fiber as we can, but it turns out that the recommended amount by FDA regulations and other sources of advice is actually really low compared to what humans have evolved with. So you normally see 35 grams recommended amount, but if you look at what our ancestors did, and they of course had different health issues than we have now, but from the perspective of the microbiota, they seem to be doing better. They used to eat maybe 100 to 150 grams of fiber per day, which is an incredible amount and definitely not something that is easy to reach, no matter how fibrous your cereals may be.
And so when you say ancestors, are you talking about hunter-gatherers living before agricultural society developed?
Yeah, it's interesting where we draw that line of, in some sense, industrialization seems to be a major issue for microbiota-related diseases. And so we normally think about most of the problems raising maybe in the last 100 and 150 years in terms of how much processed the food that we're able to get has become.
So are you saying people living like 150 years ago were getting 100 plus grams of fiber per day?
Not quite. To really go to 150 grams, like you need to go into much more ancestral societies, and even traditional societies right now that do rely on hunter-gathering are able to get that type of fiber from roots and vegetables that they source from the environment.
Interesting. And so you study the gut microbiome. We're going to one of the things we're going to talk about today. I want to go through the different parts of the digestive system from one end to the other and talk about the differences in the microbiome in each of these. But before we do that, I just want to give people like a bird's eye view of some certain things. So you hear about the microbiome a lot these days. You hear that there are I don't know, what is it, trillions of bacterial cells in our body, on the order of the same number of cells in your body that are human cells are microbial cells. And the microbiome is got some diversity to it. That diversity varies between individuals. It varies within an individual across their lifespan, et cetera, et cetera. But I want to get a sense for what the overall bird's eye level of diversity of the gut microbiome is. So in terms of the dominant phyla or types of bacteria in the human gut, what does that heterogeneity look like? Are there certain species or certain phyla that predominate heavily? Is it, very spread out? What does that diversity actually look like?
Yeah, that's a very good question. I like to think about our gut microbiota as a bit of the diversity that you might find in a forest. So there's probably hundreds of different species that are lying around, and characterizing them at the phylum level, I think is there's maybe 5 top types of phyla, but that doesn't tell you very much about how diverse that may be. The diversity of the bacteria that are within our gut is pretty incredible. If you think about what it means to be a different family, family within the world that we live in, we think about primates and felines, and so these are pretty different animals. And in the world of the microbiota, maybe we're looking at 20 different families. And so that gives you, I think, a little bit more of the sense of what the real diversity means. There are very specialized microbes that are very good at breaking down fiber, and they're just not good at all at dealing with a disruption that may be due to an over-the-counter drug that we take. There are going to be microbes that are very good at dealing with our immune system and then can hide places. There are microbes that can't tolerate the fact that in some parts of the gut there's actually no oxygen, and so they don't do very well there, and vice versa. Microbes that really like places that are not oxygenated. And so I think in thinking about it is a bit of a more of a forest ecosystem as opposed to just this catalog of microbes. It gives you a better appreciation of what the ecosystem really is and what it can do.
And so we're going to talk about like eating and how the microbiome changes in response to diet and things like that in adults. But a question I have is how does the microbiome in a human being like originally get set up? Presumably there's a microbiome even in utero, in the developing fetus and the developing baby. So where do the initial seeds of that microbiome actually come from?
Yes, this is a very controversial topic. So it's really unclear whether there are microbes in utero. There was some research that indicated there might be, but then there were some complaints that it might have been due to contamination. And so I think the jury is still a little bit out on how much it's present in utero. But the way that even if it is there, it's probably not in very large amounts because they're not easily detectable. And so the main exposure to microbes that a baby has when it comes to out in the world is really upon exposure in the vaginal tract when in the birthing canal. So that's before even the baby takes its first breath of air, the first thing it's exposed to is this coating of microbes that come from the vagina, they come from the stool. They really kind of gets all mixed in together. The birthing process is messy, and once that initial inoculation gets set up, then these are often microbes that will really help the baby be able to consume maternal milk. And it's really beautiful, in my opinion, how tightly connected this process is to the baby's health. If you think about breastfeeding, about a quarter of the maternal milk by dry weight is actually components that the baby cannot consume. They're just sugars that are broken down by the microbiota and helps it get started in a way that is healthy. So this component is so important that even mothers that are malnourished still have it. They will not have, say, all the fat compounds that are important for the baby, but they still have these sugars that are just for the microbiota.
So you said 1/4 of the weight of breast milk are sugars.
The dry weight. Mm-hmm.
Of the dry weight, sugars that only the microbes can break down.
That is correct.
So, another question there is the composition of the microbiome in the birth canal that's going to seed the baby's microbiome. You just said it's very important because that's what's going to help the baby break down a lot of what is actually in the breast milk, which is going to be the predominant food source for some time. I would imagine that the diet of the mother and other factors in her life can affect things going into her body. So diet, pharmaceuticals, what have you, could affect the composition of the microbiome that she's going to see the baby with. But I would also imagine how the baby is born could be relevant here, since not all babies are born by the traditional passage through the birth canal.
Yes, absolutely. So from the perspective of the mother, we know that having both a healthy diet as well as a healthy microbiota during pregnancy is very important, because those are the nutrients that the baby's exposed to while in the womb. And on the other side, what you get exposed to if a baby is born by C-section, say, we've found that babies tend to have a microbiota that sometimes it's even more similar to the surgeon's skin microbiota than that of the mother. And so there are now a lot of effort into trying to understand, can we actually do maternal seeding for babies that have to be born by C-section, so that you can take a swab from the mother ahead of time if it's a scheduled C-section, and then try to get this first process to happen a little bit more naturally. But maybe just to add one more thing about the importance of the mother's microbiota even before the birthing process. The reason why the microbiota is so important ultimately to our health is the fact that whatever microbes produce can ultimately affect any component in our body. So the same way that you might take a pill by mouth to affect your headache, even though you're taking it through your digestive tract, the same way the microbes, whatever they make and they secrete, can make it through your entire blood system and affect any cell in your body. And so you can imagine that we have evolved with these microbes over millions of years. And we've come to expect, in terms of our development, certain compounds that these microbes gave us. And so changes to this microbiota can potentially make an effect for a lot of different types of development. And so we're just starting to learn about some of these delicate connections that overall they're quite robust because they have come from millions of years of evolution, but we have stepped in heavily and heavy-handed in the last 100, 150 years.
And I don't want to get too off track here, but I mean, I was blown away when you said 1/4 of the dry weight of breast milk are things like sugars that the human baby itself cannot digest unless it's got the right microbes inside to do that for them. What about, like, we make so many like modern alternative foods today. When I think about something like baby formula, does it have those types of sugars in it?
It doesn't yet. This is a big race for a lot of the formula producers to try to have more of this diversity. But the problem is that this diversity of sugars is very hard to obtain, and synthesizing these sugars is not easy at all. So it's also something that you cannot get from other animals. For example, milk from cows doesn't have quite the same diversity of these sugars. Milk from primates is still different. It seems to be something that is very, very specific to the human host that we have this.
Yeah, I mean, the other thing that you said was very important, I think, is even a malnourished mother will still produce those sugars. So they must be very important for something. What are the metabolic consequences of these sugars as opposed to something like glucose or the ordinary sugars we normally think about? Are they processed differently? Are they having different physiological effects for the baby?
Yeah, absolutely. So, if you think about sugars, and even sugars that we think are not caloric for us because we can't break them down, those are sugars that usually the microbiota will break them down. So stevia, all these different alternative sugars are things that the microbiota, because of the incredible capacity of breaking down different compounds it will likely be able to draw some energy from it. And so from the perspective of these special sugars that go in maternal milk, these are very, very special things that we have evolved to produce in order to keep our microbiota together. And on the other side, if you have something like pure glucose gets absorbed and goes into your bloodstream immediately. Sugars that cannot be broken down by us, but we're basically the host of this microbiota. That's kind of how we term it in the field. These sugars will make it very far down the digestive tract. And by making it that far down, it means that they're not released into the bloodstream really quickly, which means that it's a longer term process of consumption, which is different than if you have if you drink an orange juice versus if you eat the orange, where the sugars are being liberated is in a very different place. And so this liberation of sugars, when it happens via the microbiota in distal parts of the gut, those tend to be just what we have evolved with. And so in some sense, like, they're healthy just by default because that's what our whole system is expecting to see. It's not expecting to see pure sugar within your mouth, say.
I see. So the baby's probably getting a slower drip, a more consistent intake of sugar for energy throughout the day, simply in virtue of the fact that it's got these sugars that are slower to absorb.
And also, these sugars don't get turned into glucose for the baby. The microbiota will consume them and it turns them over into other compounds. So these compounds are called the short-chain fatty acids. They're some of the major compounds that are produced by the microbiota. And they're so important for our gut that if you give choice to colonocytes, so these are some of the cells that are part of the colon. If you give a choice to these cells between eating pure sugar and eating these compounds that ultimately are turned into sugar for energy by the cells, they will actually choose to use the microbiota produced compounds, the short-chain fatty acids. The short-chain fatty acids have a lot of importance also for reducing inflammation. They are important because they're signal molecules for the brain. They have a lot of more complex interactions with our immune system and with the function of our bodies than sugar can have. Sugar is like this cheap coin that kind of gets exchanged for energy very, very rapidly. But these more complex compounds have really intricate relationship with our body function across the entire body.
I see. So many cells of the colon, or at least certain cells of the colon, you give them the choice between using short chain fatty acids or straight glucose. They will prefer the short chain fatty acids, and presumably the way to think about this would be that we've evolved for so long in the presence of enough fiber and other things to feed this microbiome, and those short chain fatty acids are produced by that microbiome, in the colon. And so those cells are just adapted metabolically to be able to most efficiently extract or use the short chain fatty acids for energy, even more effectively than glucose.
Yeah, that's really well said. And another way in which we see this really tight connection is in the fact that our body creates this layer of separation with a microbiota. And so this is a mucus layer gets created, in fact, on most surfaces, but very much so in the digestive tract. And this surface not only provides a mechanical barrier that prevents the microbiota from invading us, but it's also a source of a lot of these sugar types that are part of this mesh of this mucus component. And so when we don't eat our fiber, we're still able to feed our microbiota based on these compounds. The problem is that if the microbiota doesn't have any fiber, then it will make this layer much smaller and thinner, which then creates problems with pathogens. But in some mammals, this layer is so important because you can imagine what might happen during hibernation for some animals. Some animals are not eating anything during this time, but they still want to keep their microbiota. And these animals that hibernate have thicker mucus layers prior to hibernation that allows their microbiota to survive and actually produce nitrogen even during times when they're not being fed. And so again, like they're just really beautiful and complex interactions that we just have evolved with, and that's kind of how our whole mammalian function is expecting to have this microbial component.
I see. So before we get to the oral microbiome, the gut microbiome in the colon, we've been evolving in concert with these microbes for a very long time, and that microbiota down at that end of the digestive system is expecting a lot of fiber. And the fiber is then turned into short chain fatty acids by those microbes, and the cells of our colon really like those short chain fatty acids. They're really used to them. They're adapted to using them for energy. So I don't want to tell people there's no such thing as too much fiber, but it sounds like the dietary guidelines you said, the fiber recommendations are much lower than what humans would have been consuming for most of human history. And so they're probably well under what our cells are expecting, so to speak.
Yes, but where is the trick though. The problem is that when we haven't eaten this type of food over generations, we've actually lost some of the microbes that were able to break down this fiber. So it's not that today you and I could go out and say, like, I'm going to eat 150 grams of fiber and be okay. We probably would have really bad tummy aches. And in fact, some patients that have inflammatory bowel disease, they find eating fiber really problematic, even though it's ideally we would really like them to have more of these anti-inflammatory compounds via the microbiota. This is very much an ecosystem issue because you not only need to have the nutrient sources for the microbiota, you also need to have the right microbes to be there to break it down and to make the right compounds to do this.
I see. So over time, as humans started eating less and less fiber and their diet started to change, certain lineages of bacteria in the microbiome started dying off, and they're not with us anymore. And so if you were to just go back to eating, if you were like one of the paleo diet people and you really wanted to literally eat like a hunter-gatherer did, that would actually be problematic because you don't actually have the microbiome composition that our ancestors did.
Exactly.
Interesting. Okay, so let's now just imagine a normal human adult. And we could unpack that as much as you think is necessary, but we're thinking about adults now. Let's maybe walk people through how the microbiome looks and works differently in different parts of the digestive system. Starting with the oral microbiome. So what are the microbes like in our mouth? How are they different from microbes, further down in the digestive tract? And what are they doing for us there? Are they involved in things like pre-digestion of food? Are they doing other things? What does the oral microbiome look like?
Yeah, the oral microbiome is very diverse. It's very tolerant of oxygen because, of course, it's in an oxygenated environment. It gets constantly washed down to the digestive tract, so in some sense it can seed the rest of the gut by being there. And it's thought to break down a lot of sugars that come in from our diet. So during mastication, that's when we start breaking down the food, and these bacteria take advantage of this. And so, in fact, the mouth microbiota is probably one of the first parts of the microbiota that was identified in Antony van Leeuwenhoek in the I guess 16 and maybe early 1700s, was looking at his dental plaque and looking at how these animalcules that he called them, but that actually ended up being bacteria, how they were moving around. And so these microbes have long been known to be there, and we try to get rid of them by brushing our teeth. But in practice, they just, like any living organism, they just try to take advantage of the food resources that are available to them. They can create these strong biofilms, and so that's what dental plaque ends up being. There are these strong constructs of different species of bacteria that form these the strong and sometimes quite hard layers, which are hard to get rid of. And part of the problem and part of the interactions that, of course, we have with these microbes is what happens in bad times. So when we get caries and different dental disease, that's when there's sugar that gets left over. These bacteria can ferment the sugar. They produce acids. They make holes into our teeth. And so through these holes, they can start building up larger colonies, and more sugar gets in, and then you kind of get this tooth decay process that takes place. And again, this is something that our ancestors didn't have to deal with because they just didn't have access to sugar, and so that's not something that we're ready to deal with evolutionarily speaking.
So normally when we think about dental hygiene and cavities, we think about the practices that we have, like brushing our teeth and using mouthwash. So if someone is getting cavities, it means you're not brushing your teeth and or flossing enough. You're not cleaning your mouth enough. How much of the development of, say, cavities or something, just general dental hygiene, how much of it is to do with us engaging in those practices versus having a diet that's conducive to cavity-causing bacteria? So I guess another way to put this is, if you took a hunter-gatherer, an ancestral human, and you just fed them their normal diet and they're in that context, and you took a modern human and you fed them, say, the standard American diet, and neither one of them brushed their teeth with toothpaste. We did whatever hunter-gatherers did with their teeth. Or you took that hunter-gatherer and you just gave them the modern diet. Would they develop more cavities because they're getting more sugars?
Yeah, simple sugars that get stuck between the teeth are a major cause for cavities. And so the part of the point is that the only types of sugars that hunter-gatherers were really exposed to, and we see this in current traditional populations as well, is maybe honey, berries, and fruits. But in those cases too, the sugar tends to be sequestered within fibrous structure. So even if you get some fiber stuck in your teeth, that probably was bugging you and you take it off. And so, the old way of brushing teeth is, chewing on a stick. And so those type of things work just fine. People were not really having, as far as we know anyway, we're not really having the same dental problems that occurred when sugar was discovered and how tasty it is kind of became clear.
I see. So in the absence of food where the sugar is readily available to the microbes in your in your mouth, the old way, the traditional way that hunter-gatherers would take care of their teeth was basically just physically remove obvious pieces of food, because any piece that was there wouldn't have nearly as much sugar exposed and available to the microbes as much of our food does today.
Right. And and you can kind of see it also with pets as well. We're now having to brush our pets' teeth, which is not something that pets in the wild don't really do. The way that dogs keep their teeth clean is because they chew on bones, and that helps them keep things clean. But the introduction of also sugar, and who can resist a puppy asking for a piece of your candy, right? But yeah, this ends up being the same problem for other animals as well.
Interesting. So, as you go from the oral microbiome through the esophagus and into the stomach, how does the size and the diversity change? How does what the microbes are doing change? One obvious thing that comes to mind is when you get down to the stomach, it's obviously a very acidic environment. The same microbes won't be able to survive down there. But what does the microbiome look like in the stomach, and what are those microbes doing in that organ?
So in the stomach, the low pH is really this barrier for a lot of pathogens to get through. And so one of the things that is, for example, interesting is that for people that take antacids, this tends to be a risk factor for developing certain types of intestinal infections. And so again, like this is at the core, a big function of the stomach is to limit the infection from potential pathogens that may be coming through. But one of the things that's interesting about the stomach is that while in the main part, in the bulk, is very acidic, close to the stomach epithelium, you actually have very neutral pH. And so there's still a mucus layer, and there are microbes, notably Helicobacter pylori, which was discovered in its connection with stomach cancer. It can reside there. And so over time, there's been this discovery and this appreciation that even microbes that have for a long time thought to be really problematic, such as Helicobacter, actually have a positive effect on our immune system. So one of the ways that now the understanding around Helicobacter has changed is that it actually the best situation that maybe would be possible is that if you were exposed to Helicobacter when you're a child, but you get rid of it before adulthood, that may be the best way of setting yourself up for health. Because Helicobacter later on in life is correlated with cancer, but lack of Helicobacter seems to not stimulate the immune system in a way that is positive enough. And so this type of I think interesting interactions with the bacteria that can survive in places that are very inhospitable, such as the stomach, kind of gives a bit of a vignette into how complicated some of these interactions are. There's not going to be a microbe that is just good or just bad. Usually there's this whole spectrum of positive and negative aspects to it.
And so the stomach is this huge barrier to pathogens because it's acidic. It opens up into the small intestine, and that's where a lot of our nutrient absorption is going to be happening. I would imagine the microbiome is probably bigger and more diverse when you get into the small intestine, you start moving forward. So is that true? And, just in general, how is the microbiome composition changing as you go through the small intestine?
The small intestine is still very aerobic, so it has microbes that are able to tolerate this level of oxygen. But it's also, as you said, the site where most of the nutrient absorption comes in. And so there, the immune system goes down hard on the microbiota and prevents large-scale proliferation. So you actually have low diversity, low overall levels in an effort to try to have most of the nutrients actually being absorbed by us. And it's not until the later parts of the digestive tract that then the levels of the microbiota become incredibly high and incredibly dense.
I see. So because this is a site of stuff coming into the body, nutrients, but you also don't want bad things like pathogens to get into the body from the cavity or the lumen of the intestine. I imagine this is why. So when you say the immune system is active, I imagine that includes just a lot of immune cells that are surveilling the area, tight junctions that physically prevent stuff from getting in, all of that stuff.
Yeah, and also antimicrobial peptides. There's just a lot of activity in the small intestine to ensure that the microbial levels are really low. And when that doesn't happen, then there's specific diseases that have overgrowth of bacteria in the small intestine.
And so what would be examples of those diseases, and what are some of the general symptoms?
Yeah, so one of the probably most notable one is SIBO. So this is a small intestinal overgrowth of bacteria, and in those cases, there's some diseases that unfortunately are not very highly controlled, and one of the challenges is that our usual way, by our I mean like as a human community and human society of trying to combat bacteria when they're overgrowing is using antibiotics. But one of the issues with using antibiotics, as we found, is that you have this very non-specific way of affecting the growth of many, many different bacteria, both the good ones and the bad ones. And so the fact that bacteria can become resistant to these antibiotics has basically taken away a lot of the tools that we had initially to combat some of these diseases. And so right now the use of antibiotics around a lot of these diseases of overgrowth is changing because they're not as effective as they once were.
Interesting. Can you say more about these antimicrobial peptides? Just and briefly explain for people who don't know what exactly is a peptide, and where are they produced in the intestine? And I mean, are there? I would imagine it sounds like there's a lot of them produced, so they're very important here.
Yeah, so antimicrobial peptides are this innate way of the animal body to regulate which or how much of microbes may be around. So a peptide is a small component, small part of a protein, and they're components that they're very small and they're usually charged. They're charged in a way that they can interact with other molecules that have the opposite charge. Generally, bacteria have a negatively charged membrane, and a lot of these peptides tend to be positive. And so by attaching them to the membranes, they can disrupt them and then have this very, very blanket way of keeping numbers down. The immune system has a way of producing these that basically is like kind of a first response to changes that is not adaptive. So when we're talking about infections, there's two types of response that the immune system has. The first one is the innate response, which is the first blanket response of say, you can imagine like in a city that is being attacked, you shut down and close down all the doors. And so like it's not a non-specific way of trying to keep out the problematic parts. And so these peptides are a way in which the immune system responds very, very rapidly in a non-specific way to this type of insults. And the other one is the adaptive response, which is the one that is then more sophisticated but takes longer to actually come up.
I see. So as opposed to generating antibodies, which tend to be very specific for particular proteins from particular bugs, these antimicrobe peptides are just sort of like sticky little mini proteins that just sort of stick to the outer surface of any bacteria that might go through there.
Yeah, at a very high level. That's kind of the principle.
Okay. So you get through the small intestine. Much of our nutrient absorption will have happened now. You get to the large intestine, and it sounds like in the large intestine, you're going to have a larger microbiome, more microbes, and presumably the composition or the types of species living there is also very different.
That's right. So by the time that you get to the large intestine, all the tasty stuff that we wanted to take up from the diet, we've done that. It's out. What's left is things that we really cannot digest. And so a lot of the fiber and now that we have diets that are very high in protein and in nitrogen, there actually tends to be more nitrogen that comes through in that part of the gut. And that's also one of the factors that is just changing the way that the microbiota can function, because now we can use these more precious nutrients. But by that point, because ideally the fiber is quite diverse, there's a lot of different linkages that are connecting these complex sugars. And so you need a lot of different types of microbes that will have a lot of different types of enzymes. And so to give you a sense of the metabolic capability that the microbiota in our large intestine has, the estimates are about 150 times the number of genes than us humans have. And so like that's an immense metabolic capability that we don't want to store within us. we're basically just offloading all of this complex biochemistry to microbes that naturally have much smaller genomes, but there's many of them and many different species that can break down these fibers.
So this fiber gets down to the large intestine because we can't digest it. It can be broken down by all of these microbes. We've evolved with them down there for so long that our bodies expect that fiber to feed those microbes. They generate other things like the short chain fatty acids you were talking about. There's more to unpack there, but can you just explain for people what is fiber and why can't we digest it?
Oh yeah, that's an excellent question. So fiber is a bit of a catch-all and very fuzzy phrase. And in fact, in the microbiota field, like we've been trying to come up with a with a better terminology around it. So we often talk about microbiota accessible fiber, which is basically compounds that are not broken down by us, that are made mostly of sugar residues that are connected together with different glycosidic linkages. And so like these are different connections that in order to be broken down to get to the simpler sugars, you need very specific enzymes that will allow these enzymatic processes to take place. And we can break down some type of fiber, right? If you think about starches, we have some ability of breaking them down. So we do have some inherent ability for simple sugars to break them down. But for sugars that are more complex and that are very highly branched, we just don't have the enzymes to do this. And so that's when we really rely on the microbiota to do this.
And so like other than the short chain fatty acids, is there anything else that gets produced by the microbiome in the large intestine that we are absorbing, other types of nutrients or other types of molecules that are relevant physiologically?
Yes, an incredible amount. It's still very much dark matter. We do not know how many. We just know that an incredible proportion of the molecules that we have in our blood are not molecules that we produce. They come from our diet and they come from metabolites that our microbiota produces. And we're just starting to look at the tip of the iceberg because it's really incredible the diversity of compounds that get made. And a lot of the compounds are very small molecules that will make it to the brain. They will make it to every distal site of our body. And in trying to understand how different molecules will affect our health is a very complex question, particularly if you're to study it in humans, because every human has a different microbiota, a different diet, a different lifestyle. And so making these connections really requires careful experimentation.
Yeah, I was struck by what you said earlier too, that as the human diet has changed over time, directionally, we've been eating less and less fiber. We've actually seen a die-off of certain bacterial lineages that we used to have inside of us that enabled us to deal with that volume of fiber. So obviously the microbiome is going to change in response to our diet. I'm interested in sort of the sensitivity or stability of the microbiome. So for example, obviously the microbiome is going to change in response to what we put in our bodies food wise, but is it super sensitive? If you change your diet at all, is it definitely going to change it? Or can certain populations kind of become entrenched and sort of impervious to certain changes in the diet?
Yeah, it's both very malleable, but it's also very unique. And so there's been studies showing that you can tell who's been in a room just based on the microbial signature that they leave behind. And this is something that is pretty stable over the long term, as long as you don't do really extreme changes. At the same time, when we look at the abundance of different species, those can change and fluctuate a lot because one day you ate strawberries and that caused a bloom in a specific type of bacteria that really likes to break down the strawberry fiber. And so there's on one side, I think that it's very, very exciting how malleable the microbiota is because you can change it really drastically based on your diet. On the other side, we know that it's hard to have new species entrench. And so particularly when you're an adult and your immune system is now pretty set on who's going to be allowing to come in and out.
And so, we talked about the interplay between the immune system and the microbiome earlier. We talked about antibiotics. I would imagine that one major way that the microbiome can have big shifts that stabilize, such that someone, starts out with a microbiome, microbiome A, and they end up with microbiome B, which is just very, very different. I imagine one way that can be triggered is through the heavy use of antibiotics. So if someone, say, gets an infection and they're taking one or more antibiotics for an extended period of time, let's imagine a situation like that. The person goes on antibiotics, they get rid of their infection, they come off antibiotics. Let's assume their diet is constant. Is the microbiome likely going to go back to the way it was, or is it likely going to be different in some important way?
This is, I think, a really important topic and a really important question of kind of this recovery. There's been some studies that basically have shown that the use of antibiotics usually changes the microbiota, and particularly it affects species that are at low abundance, just because the fluctuations that end up maybe causing them to disappear, then if you don't get them back, they're just gone from your own ecosystem. And we live in a society that is very different from our ancestral ways, which prevents us from getting certain types of infections, but also prevents us from being reinoculated by microbes that we might want to keep around. And so if someone is taking antibiotics, it really depends on how long the antibiotic course is. And also it depends on the practical re-exposure that they might have to some of these microbes. And in some of these studies, they also looked at, is the microbiota that we get back the same, and are there ways in which we can help it? So one of the things that people often think about when you think about the microbiota is thinking about probiotics may be beneficial. And actually, those studies, it looked like exposure to probiotics actually got you to a different microbiota than what you started with. And the best way to get to the same microbiota that you had before was basically keeping a stock of the microbiota you had before antibiotics and then taking it back after, which of course are still very experimental ways of doing microbiota restoration. But in the same light in which how we're doing ecosystem restoration, that's a little bit how we think of microbiota therapies.
Yeah. So it makes sense. The ideal way to replenish the microbiome you had would be to keep a sample somehow of the one that you started with. Let's force ourselves to be brutally practical here, especially for the people listening that are concerned about microbiome health. So you said you have kids at home. You probably have young kids at home, it sounds like. So at some point, if it hasn't happened already, some of them are going to get sick, and they're probably going to require or be recommended to take some antibiotics. So given your background as a microbiomeologist and your role as a mother who's had to deal or will have to deal at some point, presumably, with the use of antibiotics in your children, what are the major things you're thinking about in terms of how you're going to respond during and after those antibiotics are used in terms of how you manage their lifestyle?
Yeah, so I mean, first off, I should say that I'm not that kind of doctor, so I should really not be giving any clinical advice.
I'm saying, like, right, your kids get sick, they go to the physician.
Yeah. So, I think one of the things that's really challenging about the microbiota and how we can use it in health is that humans are very, very good at dealing with acute problems. Humans are really, really bad at dealing with chronic problems. And the microbiota demonstrates itself into chronic problems, not in the acute problems. So if you have say a microbiota that is not very diverse, you may be at higher risk of developing inflammatory diseases, you may be at higher risk of developing obesity, but these are all things that don't play out overnight. And you can think of the decision that a physician has to do when there's a risk of sepsis, and the risk of sepsis is maybe 1 in 10,000. How do you weigh that against a risk of even 1 in 500 of developing obesity in 20 years? Those are the type of questions that when people are trying to use antibiotics and decide whether to use them or not, sometimes are faced with. And in general, people will tend to try to not get sepsis right now and downplay the effect, even though it might be a higher probability over the longer term. And so from the perspective of taking antibiotics, what I can tell you about, what I try to do personally is try to resist the impulse of treating something immediately. clinicians are also becoming more and more aware of the negative effects of antibiotics, not just because of the effects to the microbiota, but also because of how we're raising antimicrobial resistance that then we will not be able to fight by the overuse of this antibiotics. And so I think that overall the level of antibiotics that are being prescribed is much, much lower than it was even 5 to 10 years ago. But in practice, what I think that means is that if someone is sick, before giving antibiotics, to give it a chance to know whether it's maybe a viral infection, and then the antibiotics actually would really not be helpful. And it's also a matter of changing our perspective, also as patients, to know that some of the time the best way to beat an infection might just be to wait for our own immune system to take over and do what it knows how to do. But again, when you're a mother and you're exhausted and your kid is crying and has a fever, you want to do something about it. So they're not easy decisions.
Mm-hmm. And so let's just say, for whatever reason, a mother has consulted with a physician. Her child is sick with some kind of bacterial infection. It's decided that antibiotics are appropriate for that infection and they are used. If you were in that situation, thinking about the microbiome and the recovery phase, would you be inclined just to make sure the diet was what you thought it should be? Would you be inclined to use something like probiotics? How would you think about it in that particular situation?
Yeah, so I can tell you what I do is kind of an individual. I've had to take antibiotics. There have been times. Antibiotics are, of course, incredibly important to combat infections. And so, in terms of the recovery from probiotics, we're not at the stage yet where you can save your microbiota and then re-inoculate yourself. And so, what I try to do is to basically have a natural recovery of my microbiota by having different types of fiber. The problem with probiotics in pills is that you're inoculating 1 billion of one species. And so try to imagine what that looks like in say, Yosemite has had a massive wildfire, and now you throw in 1 billion rabbits. Is that good? Is that bad? Will the rabbits even survive, right? And so in trying to think about the microbiota is a little bit like your own ecosystem. That's also the same principles that we should apply here. If you want to do ecosystem restoration, you're probably not going to be doing very much good by adding 1 billion rabbits, but maybe you want to try to get rid of overgrowing species that may be invasive. And so you can do that with respect to your gut by starting with actually foods that are not super high fiber if you've had a tummy ache. And so step in and introduce it more slowly. So you're really trying to rebuild your ecosystem and allowing it to recover in kind of its entirety. There's also different types of probiotics. Probiotics that come in a pill form, those tend to have just one or a couple of species that in some cases may have been proven to have some positive effects, sometimes not. A lot of the ones that you find over the counter are not really proven. You don't really know whether they're the right species or not. But there's also ways of, for example, eating fermented foods, which have naturally occurring bacteria, and those tend to be very positive for stimulating the immune system. And so, unless certain things have really been shown to have like a positive effect, my family and I tend to stick with things like we'll try to have some more fermented food and try to rebuild the natural environment. And the other part of it, I think, that is in kind of an approach and lifestyle that to try to ensure that you're not blocking yourself off from any microbial exposure. That is not how we evolved with. Obviously, you don't want to go out and try to catch all the infections, but there's also no need to be scared of exposing yourself to dirt. We see this in this is the data that has been really clearly shown how, for example, kids that have dogs, kids that have older siblings, kids that have some way of bringing some amount of dirt in the house or live on a farm have much lower incidence of asthma and allergies compared to kids that live in very, very clean environments. And so there is some amount of exposure to these microbes that is just important for your immune system. And so every time that you disrupt your microbiota, that's something to think about, that in exposing yourself with microbes that are healthy for you, is a good way to try to keep this ecosystem working as it should.
Mm-hmm. That might be counterintuitive for some people. If you grow up on a farm, you have a lower rate of asthma, but aren't you around more animals and more things that could trigger, asthma-like symptoms? So is this the hygiene hypothesis? And can you just kind of unpack that a little bit more for people?
Yeah, absolutely. So this was something that was found pretty early on in settlements of in city settlements that people tend to have this hay fever when they would go visit their relatives in farms, and so this correlation of these hay fevers would then turn out to be allergies, and the fact that they were more prevalent in city settlements as opposed to more pastoral and countryside type of communities started then asking the question of, is the cleanliness part of the reason why a lot of these this type of inflammatory and allergic diseases were becoming more prevalent? And so since then, there's now been a lot of studies that have really shown that our immune system, particularly in childhood, needs to have the right types of triggers in order to work properly. And if it doesn't have exposure to some types of trigger, and these are often bacterial or dirt triggers, then it starts acting against compounds such as pollen and other types of allergens that otherwise would have not been a problem.
Interesting. So when you think about the gut microbiome, we think about obviously the gut, stuff that has to do directly with the gut, the absorption of nutrients, diseases of the gut, things like that. But the influence of the microbiome, as you know, and I think many people know at this point, extends beyond the gut, and it can affect, what our microbiome in our gut is doing can affect things, elsewhere in the body. One of the areas here that I thought was really interesting that you wrote a paper on has to do with microbial endocrinology. So can you explain what that little subfield is and start to talk about the microbiome in that context?
Yeah, absolutely. So I think this is a super exciting field because it connects the endocrine and so the hormonal changes in our body also to our microbes. And I think it really underscores how tightly connected all of the processes in our body are. We tend to think about hormones as a way that is very, very human, that affects puberty, it affects estrous cycles, it affects a lot of parts that we think that are intrinsically human. But what it turns out is that the hormones that we produce can be modified by the microbes in our gut, and they can change either by activating or deactivating them. So to give you a very high level summary about this, in either the testes or in the uterus, there's hormones that are produced. And so for females, we mostly think about estrogens, and for males, we mostly think about testosterone, but these are produced both by males and females. And when they're produced and they're given out into the blood, they usually come associated with some specific proteins that would prevent them from actually becoming active. And there's also some modifications that happen in the liver that inactivate these hormones. So the body is constantly turning off these hormones and the way that they act. So while normally when we go, say, get a blood drawn and we look at these hormones, we normally take a look at the overall levels, but it's not always that you'd be looking at the active levels, which ultimately are what make a difference. And so while the liver continuously inactivates these hormones and shuttles them in a way that will make them more likely to go out in urine, a lot of gut microbes actually do the opposite. They will take away this inactivating molecules and will activate them again. And so you have this balance in which your microbes can activate them. Some microbes can even degrade hormones like testosterone. And you have this balance in which now your body is responding both to cues that come from our own cells and cues that come from the microbiota in order to change these processes.
I see. So the microbes can directly. It sounds like they can directly modify things like sex hormones.
Exactly.
Are there any things? Are there metabolites they produce that have estrogenic or androgenic effects. So, for example, are there certain components in food that are eaten up by the bacteria and causing them to spit out something that has a hormone-like effect in us?
Yeah, so this can happen in a number of different ways. Like there are soy-rich products that can be metabolized into compounds that look like estrogens, and so like these can get cross-react with estrogen receptors and give some of the same effect with with estrogen.
So you said soy-rich products?
Yes.
Okay.
The other thing that can happen is that there are some microbes that can produce their own hormones, particularly in the case of testosterone. And they can do this starting from cholesterol in a very similar way to how, as humans, we use cholesterol to get down to hormone creation.
Do we know the net effects of any of that? So, for example, soy, like soybean oils, things like that, very common. They've become much more common in recent years than they have in the past. Is that having like a net estrogenic or anti-estrogenic effect through some kind of competition between these estrogenic metabolites and our endogenous hormones?
Yeah, that's a very good question, and I think a very complex one. I'm not aware of, I guess, of a full agreement about this. That there are human populations that have consumed soy for millennia, and in as part of their normal diet and their normal cycle. So I don't think that a blanket statement would be appropriate, but it really very much depends on how different microbiota are balanced. It also depends on how the diet may be balanced with other compounds. But there's definitely some of the data that I think is very strong in showing the impact of the microbiota and modifying these hormones is that when we look at animal models that do not have a microbiota, so these are we were talking about that with in humans, in mammals anyway, in the uterus, there's not thought to be a microbiota. So there's animal models that can be derived by C-section and do not have microbes anymore. And so these can be a way in which we can test like what is the effect of not having a microbiota. And so in these animal models that don't have a microbiota, but have an otherwise normal function and normal diet, we see that they have very strong changes in their puberty, in their sexual function, and so this underscores the importance of the microbiota in having some of these changes to hormonal health that are normal.
I see. So it sounds like most of the details around all of that have yet to be worked out, but we at least know that if you take away the microbiome entirely, you see hormonal and other effects that are strong enough to change things like when puberty happens. So pretty major developmental changes.
Yeah, and some of the other studies that have been done have been, for example, around prostate cancer. And this is a disease that often there's imbalances and maybe too high levels of testosterone. And one of the things that sometimes is done in patients that have prostate cancer in a way that where there's too much production of testosterone is that there's a castration that takes place to try to reduce the testosterone and limit the impact of the negative impact of the testosterone. But there have been some patients that were found to be basically insensitive to the castration. They were still producing very high levels of hormones. And in those cases, the microbiota was implicated in this high production of testosterone. And so the mechanisms are still being worked out, but there's now starting to be a lot of data that implicates the microbiota, both in positive but also in negative ways in this changes in hormonal and active hormonal levels.
Are there any connections between the microbiome and gut hormones that regulate hunger itself? So another way of saying that maybe is, can changes in our diet or changes that we need to make to our diet for natural reasons, like, I could imagine, if a woman gets pregnant, for example, her caloric and her nutrient needs are going to change. Are there any changes there that seem to be mediated by the microbiome?
Very much so. So this is an area that I'm tangentially informed about, so I'm going to be pretty general about it, but there's definitely evidence that the microbiota can influence hunger. They can influence how the feeling of satiety, and so the microbiota is so tightly related with us that it is influenced by our circadian rhythms. So circadian rhythms mean when the expectation that our body has of daytime and nighttime and of cycles of feeding and things like that. And so one of the things that anyone that has ever traveled and gotten jet lag knows is that there's definitely discomfort and changes to your digestive health and the way that you digest things. And that's partly due to the microbiota not knowing anymore what's going on and expecting food at times that it's not coming. So there's definitely changes that you can kind of experience in your day-to-day life that have to do with the microbiota and the interaction that it has with our expectation or when food should be coming or how hungry we are.
So obviously we've talked a fair amount about how what we put in our body changes the microbiome. We focused largely on dietary inputs. We talked about antibiotics as well. What about things like pharmaceutical medications? What do we know about, anything from, psychiatric medications to say, hormonal medications like birth control? I would guess that probably anywhere you look, there's going to be some effect. But what are some of the most salient things that we've learned or that we already have some handle on in terms of other medications besides antibiotics that we put into our bodies fairly frequently that are having major changes in the microbiome?
This is, I think, a very important and a growing area of research. So anything that makes it through our gut obviously can be modified, even broken down by the microbiota, but even things that don't start out in our gut can make it there. We talked about hormones and how they can be modified in the gut. Anything that makes it through our blood system can potentially be modified in the gut because of the high interaction between the portal blood system and the gut and the exchanges that can happen there. So there's definitely a lot of effort in trying to understand how different microbes can either enhance the effect of certain drugs or prevent them. So to give you an example, there are some drugs that can be broken down by the microbiota, and so they don't have an effect anymore. There are drugs that we deem safe but they will have a negative effect on the microbiota over longer timescales. So to give you a sense, a lot of the over-the-counter drugs that we take, we talked a little bit about antacids, like this is true of laxatives, they will just change the microbiota because they set up an environment that is not the same anymore. So laxatives often work by creating an osmotic imbalance with the gut, which draws out water and increases the motility, while it also draws out water from the bacteria. And some bacteria can't tolerate this, and so they will go away after a long-term exposure to laxatives. And the problem, though, is that it doesn't, at least to me, it doesn't make sense to say like, well, we shouldn't be using any drugs. Like that's obviously not the point. But I think it's important to know that just like with any ecosystem, if you apply a change, there will be a response from the ecosystem. And knowing what's good, what's bad, what's neutral, those are very loaded words for ecosystems, because if you think about a forest, you can probably distinguish a forest that is healthy from one that is unhealthy, but it's hard to know whether a forest is healthier than another one if they both look lush.
Yeah. And even if you can easily identify healthy versus unhealthy when they're clearly different, that's very different from saying, so you can't just look at a healthy forest and be like, oh, it's got X, Y, and Z. So if we put those things in the unhealthy one, it will become healthy. It's exactly. So complex in like literally the math sense that you just can't predict system level outcomes from individual changes and individual variables.
Absolutely. And so, in thinking about microbiota changes, say that you want to take, as you said, if microbe X is good in you, it may not be good in me because I don't have the rest of its friends that would keep it happy, right? And so the way that this kind of applies is, I think they will have to play out on a much more ecosystem level type of health approaches.
What are some of the major projects and questions you're working on in your lab right now?
we're very excited about thinking about the microbiota at different scales. So from a more clinical perspective, we study a lot the impact of the microbiota on inflammatory type of diseases, for example, IBD. So Crohn's and colitis are some of the diseases that we're looking at, and we're connected with clinicians to try to understand how changes in the microbiota and how specific types of bacteria that can cause inflammation, how those change the environment and then lead to a microbiota that is less resilient. Then scaling this down to specific applications, we're interested in how certain types of drugs may affect people that have different types of microbiota. So to give you an example, what happens when people undergo bowel preparation? That, of course, reduces very strongly the microbes that you have there. What happens if you lose some of the microbes that are important? And so we're focusing in on specific microbes that we know that are being lost in industrialized human populations and trying to understand, do we want them back? If we do want them back, can we get them back? How do they change our immune system? How did it change the interaction between different microbes? And so, in kind of scaling from clinical questions, we kind of go down to the individual cell level and trying to understand how are these microbes producing what they produce, how are they interacting with other microbes, and how can we strengthen some of the relationships in a way that as we proceed towards microbiome therapies, that we have something that, talking about ecosystem restoration, that we actually can make useful for the human population as a whole.
So one of the things tied to the microbiome that I've heard about that where you see very dramatic results, types of results where they write about in the newspapers and stuff, has to do with fecal transplants. So you perform a fecal transplant in animal models, for example, and I believe that they've seen results like, if you do the transplantation from the skinny mouse to the obese mouse becomes skinny. Dramatic results to do with diseases of different kinds. So why do fecal transplants seem to produce results that dramatic? And what are maybe one or two of the most well-known or well-worked out examples there?
Yeah, so the fecal microbiota transplants have been demonstrated particularly in animal models, but they've also been shown to be incredibly helpful in combating C. diff infection. So C. difficile is this pathogen that often comes up after antibiotic treatment, and then it cannot be eradicated because it's very resistant, and in fact it thrives when the rest of the microbiota is depleted by antibiotics. So it really takes advantage of a reduced diversity of the microbiota. And so in early experiment was tried in cases in which patients were really struggling, and it was kind of deemed to be as a last step and a last try, was to take a microbiota from a healthy donor and then transplant it. And in those cases, the efficacy of this treatment was incredible, over 90%, which is something that is very rarely seen in any type of therapy that we would normally deal with. And so this led to this very strong excitement of how we could restore these disrupted ecosystems by transplanting these healthy microbes. And one of the challenges with this is that, as we talked about, these are complex ecosystems which are very, very diverse and very, very messy. And so one of the ethical questions around it is that what constitutes a healthy microbiota that we would put into a patient that has a specific type of disease, also with the understanding that, say, you may have a very healthy tropical forest and you're trying to transplant it into a desert. Well, it probably will not take hold there because you just don't have enough water to support it. And so in more complex diseases where this has been tried, the efficacy is much lower, and we find that a lot of the microbes are not actually able to survive when they're implanted. And so this is, I think, something that we need to go much more towards precision medicine for this type of approaches, because right now it's a very blunt tool of just saying like, well, we'll take this ecosystem that seems to be healthy in this one person, put it in a completely different person, and hope that it provides the same types of health. So probably the C. diff example is probably one of the largest examples. In animal models, there's a lot that has been uncovered, both from the perspective of what is transferable. So there's definitely, as you mentioned, the obesity that can even be transferred, because sometimes also the opposite experiment was done in which an obese microbiota was transplanted into a skinny microbiota, and the skinny mouse became obese. And it turned out that the reason why that happened is because the microbes from the obese mouse were very, very good at capturing energy. And so they were releasing a lot more energy for the same amount of food to their host than the microbes coming from the skinny microbiota. And so that there's going to be a lot of things that are transferable, and some things that maybe are not. And so, this question about transferring obesity is one of the things that a lot of patients worry about and want to know about their donor, whether this may be the case. But in terms of the human data, is still things that are really very much being worked out.
Mm-hmm. So yeah, the microbiome has captured popular consciousness to some extent, to a pretty large extent at this point. I think one of the reflections of that is that it's very common to see today people taking probiotics. And I don't mean like someone has a specific deficit, like they had antibiotics and like they know for a fact their microbiome has been depleted. I don't even mean people that have like actual gut issues that have been diagnosed, but just normal people walking around that are just sort of concerned with their health at a normal level. They've heard of the microbiome, they've heard of gut health, they see probiotics of all kinds marketed out there on store shelves. I know a lot of people in my life that just take a probiotic when they take, a vitamin C pill or a vitamin D pill or whatever that's part of their daily supplement routine. And as far as I can tell, most of the time, they don't even know maybe what the species are that are in there. They don't really know what's known about them. Taking a sort of a bird's eye view, but thinking about the average consumer out there, the average person, what are your thoughts on the probiotic products that are out there? Do you take any? How should people think about that? Do we know enough that it even makes sense that people are buying these things? What's your take on the probiotic supplement industry?
I think we're very much in the early days, and it's an industry that has very much been exploited because of the excitement that people will have around diet and around supplements. And so we've tested several in the lab, and we found that sometimes they're not alive. Often they're not the species that they say they are. And so the ones that are not regulated tend to often not be what they're marketed to be. That said, they're unlikely to do harm because they do tend to be bacteria that are easy to be grown, and they also tend to be not the type of bacteria that adults would have, which is one of the parts that I think that is most bizarre about the use of probiotics, is that they tend to be bacteria like lactobacillus, bifidobacterium, all of these bacteria that are thought to be mostly, or they're found mostly in babies, not in adults. And so there's a little bit of a disconnect about how relevant they would even be with an adult taking the supplement, which is usually the case when that happens. There are probiotics that have shown efficacy in human studies for very specific indications. But in those cases, I think it is important to try to see what they're being used for and how they're being used. One of the challenge with microbiota therapy is that oftentimes, even when we want a microbe to stick around and we provide it in a probiotic form, it will actually just transit through. And so that's part of the idea with saying that for the most part, they will cause no harm. But one of the challenges is that even if you do find that microbe that you really, really want, it's maybe not something that you're able to have and stick around. And then conversely, there are microbes that, as I was saying, for specific indications have been shown to be very important. And an example of this is E. coli Nissl. And so this was a microbe that had been found in 1917 during World War I, when there was like this outbreak of salmonella, and there was like this one soldier that had shown that it was resistant to this, and it turned out that it had this microbe that basically was providing colonization resistance from this pathogen, just based on the fact that it had a similar way of nutrient requirements than the salmonella did, and so basically displaced it based on this. And so there are some microbes that have been very well worked out for how they work and how they could protect from specific things. But we're not there for most of the ones that you would see in in stores
I
at
see.
this point.
So it sounds like a fair summary of your perspective for the average consumer is that a majority of the time, a healthy majority of the time, the casual consumer, someone who's just concerned about gut health, but doesn't have a super specific thing with a super specific diagnosis where a particular species of bacteria is known to be the solution. The average consumer probably not worth it to stress about taking a daily probiotic.
Again, with the caveat of not giving medical advice, it probably doesn't make the big of a difference. But I think that for all things that have to do with health and diet, we're not there yet at the level of looking at one person and being able to say, these are the bacteria that you need. This is the diet that you should have with these bacteria. We're getting there, where we're not there yet.
Mm-hmm.
So I think that there's an important space for people to try different things and see what works for them.
Mm-hmm.
Because sometimes there are things that are unexpected. Has happened a lot with, for example, with IBD patients that they have found that certain types of food that normally would be counter indicated in general for patients with inflammatory disease actually have found that help them. And so I think that there also needs to be some openness from both the scientific and clinical perspective that there are a lot of things that we don't know, and even sometimes traditional medicines that we've discounted because in some types of trials have not worked. They may work for individuals, and so there's so much diversity, not only from the microbial side, also from the human side, that there will be things that are not standard that work for some people, and I think it's important to be open to those as well.
Can you actually, just give us the basics on what IBD is, and how common it is? I get the impression, just sort of walking around and hearing about it from people, that it seems to be becoming more prevalent. So is that true? And what exactly is IBD?
Yeah, inflammatory bowel disease is an inflammatory disease that, as the name says, involves the digestive tract. And one of the things that I think is most critical about inflammatory bowel disease is the level of pain and effect on the daily life that it has on patients that have it. It's an extremely challenging disease that it's invisible because it's not something that will be overt. And so people that suffer from this disease are often feeling unseen, and they're it's also because of affecting the bowel is something that is hard to deal with and explain when sometimes like you might have to go to the bathroom really quickly. And the prevalence is quite high. It's almost at 1% in North America. And it's by 2050 it's expected to be at 1% of the population, which is it's an incredible fraction of the population. It's also being diagnosed earlier and earlier. There's a lot more pediatric patients than there used to be. And one of the things that I think is really interesting about inflammatory bowel disease is that while it has a genetic component that is still being sorted out, and this is a pro-inflammatory response that the immune system has, and effects on the intestine specifically. The genes that are involved with this are actually genes that have been known to be around human communities at least since until the Black Plague. So there was recently a study that showed how some of the genes that helped protect us against the plague in the medieval times were actually selected upon, and those are genes that are known to be positively correlated. So they seem to be at least enriched in people that have IBD. And one of the things that for a long time scientists wondered about is that, well, if these genes have been around for so long, why are we only seeing IBD now? Why is this a modern disease?
So just to make sure I'm following, you're saying that there's genetic evidence that people who survived the Black Plague because they had a genome that enabled them to survive the Black Plague, their descendants are now more abundant than the descendants of people who didn't live through that plague and pass on their genes. And those same genes seem to be connected to IBD.
Exactly. Yeah. So those genes tend to be, when enriched, they tend to correlate with the development of the disease. The thing that's interesting too about IBD is that while those genes have been present since at least medieval times, they the first diagnosis of IBD did not happen until the 1800s, and also it only happened in industrialized countries. So when we look at, for example, in certain countries in Asia, the first diagnosis of IBD has happened much more recently and really strongly correlates with industrialization. So it correlates with changes that have to do with increased use of drugs, increased use of foods that are highly processed, and the changes in lifestyle that we kind of associate with industrialization itself. And that's really where the microbiota plays in a role, because while our genome has stayed relatively stable over this time, one of the things that has changed very much has been our microbes. And what we see in patients with inflammatory bowel disease is that they tend to have much lower diversity of the microbiota, and they tend to have a microbiota that it's pro-inflammatory. So instead of having these microbes that produce these anti-inflammatory compounds that we were talking about earlier, these short-chain fatty acids, we actually see that they produces compounds that tend to be pro-inflammatory and tends to have species that can cause inflammation throughout the body.
Interesting. So when you talk about fostering a healthy giving the microbiome what it needs to thrive and be a healthy microbiome, relatively speaking, the thing that you talk about most there, in terms of dietary components, is fiber. So we've talked about that. High fiber. A sufficiently high fiber content in your diet is important for the microbiome because it's a primary food source for the microbes that are living in your large intestine. Are there any, you talked about fermented foods as well. So fiber, fermented foods, those are the ones I hear about the most. Are there any other big ones in terms of nutrients in our diet that are good for the microbiome? And then the flip side of that, are there any things in the diet, any particular components that tend to be good for a not so healthy microbiome?
Yeah, so other things that I think that in general, having a diet that is diverse and has enough of the nutrients that you think about being healthy for the human tend to be also healthy for the microbiota. The fiber part is definitely one of the major sources of nutrients for the microbiota, but the microbiota also requires a lot of the same nutrients that we otherwise would think about, whether it's making sure that there's nitrogen. One of the things, for example, in the gut is that there's a very high shortage of iron, and the body does this on purpose not to have overgrowth. So, anyone that has tried taking a lot of iron supplements like may suffer from constipation through the microbiota being able to overgrow. So there's a lot of other nutrients that we usually don't think about that will affect the microbiota through this. On the other side, the nutrients that may have a negative impact on the microbiota as it relates to our own health are things like emulsifiers. They can negatively impact the microbiota because, for example, when you're taking emulsifiers, so these are compounds that often are used in foods to hold them together instead of having them separate between the oil parts. So you can think about salad dressings and things like that have a lot of emulsifiers. And those are things that work a little bit like soaps. And so when you take them, they will also wash things off in your intestines. For example, the mucus layer that we were talking about that creates this healthy barrier between us and the microbiota, and also can disrupt the membranes of the microbiota. We talked a little bit about alternative sugars and how those ones can be cheap sugars for the microbiota. They will still give us energy, even though they're technically not taken up by our own cells directly. And so there's a lot of different compounds that can have these effects that are indirect and they can negatively affect our health.
I see. So emulsifiers are one because they literally wash away some of the microbes in our gut.
They will disrupt surfaces that have some fats and associated with them. So again, these are kind of indirect effects, but they can change the membranes of these microbes and take off some of our mucus protective layer.
I see. So it's altering the gut environment in a way that's then going to naturally lead to different populations thriving or dying off.
Exactly.
Interesting. Any other major ones that you can think of or that have been clearly demonstrated?
The in practice, anything that we take by mouth will change the microbiota, and knowing whether it's good, bad, or neutral, as we were saying, is going to be challenging. One of the things that I think that it's interesting in thinking about more obvious things like taking antibiotics is that the microbiota itself produces antibiotics all the time, and in fact, a lot of the antibiotics that we have are produced by the microbiota. The problem is not the compound. The problem is the amount and the location. When microbes produce antibiotics, they do it to have very short range interactions with microbes around them and keep those at bay. But when we take antibiotics, we take them and we flood the entire system with super high concentrations of them, which are then not natural. Things like vancomycin have been in the repertoire of antibiotics of our Earth for millions of years, and within 30 years we were able to ruin it and make it become not successful anymore, because now there's resistance. And despite this millions of years of use of this antibiotic by the microbiota, the microbiota used it so locally that it did not give rise to resistance. And so that's going to be happening similarly to other compounds too. One of the issues that we have with the way that we take compounds by mouth is that we tend to flush our system with very high levels of things, like even contraceptives and other compounds and medicine that we take by mouth. If only we had a way to have them more localized, they could have a very more limited impact and have different effects and impact the rest of our ecosystem in a way that is less major.
Yeah, that makes sense. If we are taking medications and other things systemically, oftentimes the benefit or the thing that we want to happen is a more local thing. It's happening, in particular cells or particular parts of the body. So if you had a local delivery mechanism, you would naturally decrease the side effects that are going to come from that thing going elsewhere and just doing other stuff.
Yeah, and I think that is also one of the reasons why the microbiota is so exciting as a therapeutic, and this is something that my lab also works on, is trying to see, can we leverage the microbiota as a way to have a very specific delivery system? So now you can think about these microbes, they sense their environment, they're able to tell when, say, there's changes due to inflammation, there's changes because maybe cells are growing faster, and so the temperature is higher because there's a cancer there. Those cells can detect these changes in the ecosystem. And if the detection of those changes was tied to the delivery of specific drugs, then now you have drugs that are carried by these microbes that can be delivered only at the site where they need to be delivered. And so like this delivery of drugs through the microbiota is one of the frontiers that we're thinking a lot about in the next stage of therapeutics.
What's one good example of a major unanswered question to do with the microbiome that you think we will likely have solid answers to in the near future, meaning, let's call it 2 or 3 years?
A really major question that I think that we'll have answered is how the microbiota can insert itself. So, we were talking about from the perspective of the probiotics, like it can be really important. If we want to restore species, can we get them back? And so this question of when are we able to get them back, I think is going to be a major one that will be answered. On one side, we may be able to do fecal microbiota transplants of our own microbiota to restore it, but what if we wanted to take microbes that we have never been exposed to? Do we need to do this in infancy, or can we do this later? I think this is a very important question, and I think it's one that we'll be able to answer in the next few years.
I have not looked into these in detail, but there are now companies out there selling products where you take a sample from your mouth or from the other side of your body, and you send it in, and they analyze it, and they can give you a fingerprint of your microbiome, and apparently give you, diet recommendations and things like that based on those results. Do you think those are legitimate? Is that tech there? Is our ability to do that accurately in place? And are those things that could be worth looking into for those interested?
There's starting to be some studies, particularly out of Israel, that are correlating the breakdown of specific fibers and sugars with specific microbiota makeups. But I think we're still in pretty early days. So right now, if you were to phenotype your microbiota, you basically would get a list of species. And so think about what that would mean if you were looking at this species in a park, right? Like there's 50 lions and 73 gazelle, and right, it's not that informative. It doesn't really tell you about, are the lions healthy? Are there enough gazelles? And so we're still at the stage of, by looking at these list of microbes, there's only so much information that you can have. Like you can know that maybe there's no elephants there, and actually I think that for your environment you really should have elephants. But we're not there yet at the level of having this phenotyping be useful enough, except in extreme cases, right? Like if you have a lot of invasive species, then it becomes obvious that something needs to happen. But again, like if you have something that looks relatively healthy, we still need more data to know you should be eating more broccoli and have more bacteroides in you. We're not there yet.
Well, I mean, we've gone over a lot. Based anything to do with the microbiome, anything that we went over that you want to reiterate, or any final thoughts you want to leave people with, given the topics that we touched on today?
Yeah, so I think that the main thing is that it's a really exciting time for the microbiome. I think that there's this renaissance and appreciation of the importance that it has. I think it's a little bit like it was in the early 2000s for the human genome, right? Of the I think the promise that it has, and for revolutionizing medicine. And one of the challenges with the human genome is that the people got so excited about it that then they got a little bit burnt out because then some of the impacts were not seen right away. You know, gene therapies, we're starting to see it now, 20 plus years later. And so I think that we're a little bit in the same phase for the microbiome, is that there's sometimes a bit too much hype in that it does affect things throughout the body, but in terms of seeing solutions for how to get ourselves be better, we'll need a lot more than saying, here's your pill and you'll be fixed. So in thinking about these, I think that the main thing that is important is to think about them as big ecosystems that we carry around, that we should take care of and try to make sure to reestablish and feed and keep diverse. But they're not things that are likely going to be fixed by one pill at any point, because just like with gardens and with forests, there needs to be a good amount of attention and care for them throughout the diversity of species and interactions that they have with us.
All right. Well, I think that's a great place to end it. Thank you very much again for joining me. This was interesting. I've covered the microbiome before, but we got to stuff today that was very different than anything I've talked about with other people. So thank you.
Yeah, thank you so much for the interview. This was fun to chat.